Hello students, I'm going to make a video here for you today that will serve as both the makeup video and also a little bit of a review video because I'm going to back up just a little bit from what we talked about in class, and I'm going to make sure that you understand um, the way that we got to working on our new Sesame Street genetics characters. And then we're going to talk about the Sesame Street genetics character, uh, the new character worksheet that you were supposed to do in class. So, um, the first thing that I want to go through is how would I take two of the Muppets, cross those two Muppets, and wind up with a new character. And so the easy way of going through this, instead of going all the way through the process of meiosis um, and looking at how each individual gamete is formed, is that we would just take a look at the chromosomes that each of the parent organisms has. And then we would pick out one of those chromosomes. We'd flip a coin to see which of those chromosomes out of each set is passed on into the gamete or into the sex cell. And then we would see what happens with the offspring. So let me talk you through this with Abby and Grover. So I'm using the original Sesame Street genetics packet that we worked on the whole week before fall break in order to figure out what the genotypes are for each of my characters. So for Abby, we filled her out together as a group. You can find her genotype and phenotype in I think it's the day one or maybe the day two slides presentation, but you can also find your whole setup in the Sesame Street Genetics packet. So that packet has figure 2 in it. Figure 2 in that packet has all of the Sesame Street characters. So it's Abby, Grover, Zoe, Elmo, Telly, and Hunker. So it has all 6 of those characters, and you are supposed to fill out the genotype and phenotype of all of those characters. If that's not done, then you'll need to go back and do that because this part of the activity doesn't work if you don't have that done. So make sure that that's done and you're going to refer to it. Now, I'm going to look at Abby. So in all of Abby's cells, except for her sex cells, so except for the eggs that she'll produce, in all of Abby's cells, she's going to have 2 copies of chromosome 1, she's going to have 2 copies of chromosome 2, and she's going to have 2 copies of the sex chromosome. I have made these chromosomes different colors simply so I can keep track of them. There's nothing magic about the coloration. I just made different colors. So here's Abby and here's Grover. He has 2 chromosome 1s, 2 chromosome 2s, and 2 sex chromosomes as well. So again, nothing magic about the colors. I use the same colors as over here so that when I flip a coin, um, when I wind up with either heads or tails, I'm going to know which color chromosome to put into the gamete. Now, I don't actually have a coin in front of me to show you, so I'll just use my can of hand cream here. So here's the heads, here's tails. All right. So when I flipped for Abby for the first chromosome, I ended up with tails. So tails tells me to choose the purple chromosome. So the purple chromosome is what's going to be passed on. The information on that purple chromosome goes right into the egg here. Same thing here for looking at the sex chromosomes. I flipped. I got heads. So this over here tells me that I'm going to pass on the black chromosome. So there it is. And for uh, chromosome 2, I got tails, and so that told me to pass on the brown or maroon or whatever color that is. And so the information, the genetic information that we have up here on that brown chromosome, well, that's the information that's going to be contained in this egg. This chromosome would go into a different egg. This chromosome is going to go into this egg. So that pink chromosome, the green one, and the red sex chromosome, those are going to go into a different egg, but they're not the one that's going to be fertilized. Same process happens here with the sperm. We've got a tails for the first one, and then we've got heads, green for the chromosome 2, and then tails again for the last one. So for this, we're going to have the Y chromosome. Now, the Y chromosome does not have the hair type on here, so there's no gene for hair type that's being passed on from Grover. He's not contributing that at all to the offspring. So then I'm going to take these 3 chromosomes and put them into our new offspring. These 3 chromosomes in here, because when the egg and the sperm join together, this is what you get. They mix up their genetic information, and now we have this new organism down here. So I'm going to take a look at this organism, and I can see that I've got um, a recessive allele and a recessive allele. So I can write that information over here. There's my genotype for the baby. And then I can look back at the information in my packet, and I can see that this genotype will result in this phenotype. So the nose, homozygous dominant, that gives me pink. The lips, heterozygous, that gives me red. The body is capital B, capital O, that gives me magenta. And then the hair, because we have an X chromosome that has the hair type on it and a Y chromosome that doesn't have any information about hair type, I have XH because that's what's contained on the X chromosome, and then Y. And it doesn't have an H because it doesn't have the gene. The X and Y tells me that this baby is male. So here's a little baby boy Muppet that has flat eyes, a pink nose, red lips, magenta body. It has a hairy head, so it has like the little, fuzzy stuff on its head, and it's a boy. Okay? Now, I would take this information right here and I would just put that information directly into this right here. So my genotype and phenotype I just did, if I take a look at this, my genotype and phenotype are right here. There's my genotype for the baby, there's my phenotype for the baby. So I would just put that information up here, and then I'd go up to the information that I have for Abby. And so on this, I would just type in for looking at Abby, I can look at her right here, or I can copy this information from the figure 2. And so I've got a recessive allele and a recessive allele, so homozygous recessive. So I would type in right here, and it'll correct it for me. 2 little e's. I'll have to go back and fix that little e. And so she would have, she has flat eyes. Okay, so that's how I would fill this out. I would look over at Grover. He also has 2 little e's. That's flat. Again, autocorrect happens here, so check that. And then for the offspring, 2 little e's, flat. Okay, so this is how I'm going to fill out this part of my Sesame Street Genetics new character worksheet. Okay, so I'm going to take that information out, and then I'm going to run through what we talked about in class today. So we talked about like we had different people who crossed Zoe and Elmo. And we had them share out their information. So as we went through it, not all of the people who crossed Zoe and Elmo got offspring with the same eyes. Some had flat eyes, some had Google eyes. Not all of them got the same nose color. Some had pink, some had, I think it's orange. Not all of them had the same lip color. Not all of them had the same body color. Not all of them had the same hair type. And not all were boys, not all were girls. So we could see within our class that we had genetic variation, even though those people used the same 2 parents. So that started our conversation as we talked through how to answer these questions. So, what I asked the class to do was to look at their offspring. Was that offspring genetically identical to either the mom or was it genetically identical to the dad? We had several that said it's close. Some of the things were the same, but I'm not sure that anybody in any of my classes said that yes, it was completely and totally identical. So then we went to, was the offspring phenotypically identical to either the mom or the dad? And again, no. We had one student who the boy offspring looked just like the mom, like all of the phenotypes were the same, except it was a boy instead of a girl. So that was a little different. And we talked about how with 5 different characteristics, that's a possibility. But what if we have 50 different characteristics? How likely would it be to happen? What if we have 500 different characteristics? So 500 genes with all of their variations. How likely would it be that you would get a genetically identical offspring and parent? How likely would it be that you would get a phenotypically identical offspring and parent? So they would look exactly the same. Probably not too likely. What about if we had 5,000 genes? Well, now this is starting to get like, I mean, I guess there's a chance, but that's going to be a really small chance. And then we go on to talk about that in the human body, we have about 21,000 genes. So that's a whole lot of genes. And, it's going to be super, super, super unlikely for an offspring to be identical phenotypically to a parent, and almost impossible for an offspring to be genetically identical to one parent. So we said, okay, we get this idea. This is not likely that we're going to find that an offspring is genetically identical to the mom or dad, and not likely that we're going to find that an offspring is phenotypically identical. So it's not going to have the same genetics, the same letters. It's not likely going to have the same phenotype or physical characteristics. So then we moved on down to number 4. And so here for number 4, this tells us to use our model. So there are a couple different models that you can use. You could use a complete model of meiosis. If you're watching this in VLA, if you want to learn about meiosis, that's in the enrichment for the Wednesday before fall break. I think that would be September 30th. Um, or you could use the kinds of things that we have done withdrawing these withdrawing the things out. Let me pull this back up. Withdrawing the things out here with Abby and Grover. So you could use any of these models that we've used. Probably the easiest model to use is this one right here. Okay, so moving through this model and going all the way down to where you create your offspring. That's probably the easiest model. And so here are the key points. This might not be quite exactly how you would describe it, but this would be information that would need to be contained within your explanation. So, gametes form that carry only one copy of each chromosome from each parent, and you can see that. You can see that right here, as there's one copy of chromosome 1, one of the sex chromosome, one of chromosome 2. Over here for Grover, one chromosome 1, one chromosome 2, and one sex chromosome. So gametes form that carry only one copy of each chromosome from each parent. Independent assortment means that there can be multiple combinations of chromosomes in the gamete. So independent assortment, I'm going to highlight that, independent assortment, that is the kind of an important term here. That means that when we flip the coin for chromosome 1 and we flip the coin again for chromosome 2, the outcome for chromosome 1 didn't affect the outcome for chromosome 2. The outcome for chromosome 1 did not affect the outcome for chromosome 2. So whatever I flipped here for chromosome 1, if I got heads, that did not mean that I automatically got tails here. It didn't mean that I automatically got heads here. I had a 50-50 chance of getting either one. And that is the same principle, even though, clearly our cells are not flipping coins, but that's the same principle that occurs within our cells when our gametes are being formed. Chromosome 1 sorts out, chromosome 2 sorts out, chromosome 3 sorts out, and that goes all the way down through chromosome 22 for humans, and then chromosome 23 in humans is our sex chromosomes, and those do the exact same thing. So we've got an X and a Y if we're male, we've got an X and an X if we're female. So um, those are the ways that we see independent assortment showing up here. So independent assortment means that there can be multiple combinations of chromosomes in the gamete. So Abby could have had the purple and the green and the red, or the purple and the green and the black, or the pink and the green and the red, or the pink and the green and the black, or the purple and the maroon. And you get the idea. I could go on and on and on. So there's a bunch of different combinations that you could get just from looking at 3 chromosomes. Imagine the genetic variation if you expand that out to 23 chromosomes. There's a lot of genetic variation. Okay? And then the gametes combine to form the new organism. So if we look at that on here, we've got the egg and the sperm. So the egg here and the sperm here, they are combining together to get a fertilized egg. That's called a zygote. This is the beginning of our new organism right there. So the gametes combine to form the new organism. And this resulting zygote, so this resulting fertilized egg, has a combination of chromosomes from the mom and the dad. So here's from Abby, there's from Grover. This one is from Abby, that one's from Grover. This one's from Abby, that one's from Grover. So this is a combination of the genetic information from Abby and Grover, but it's not all of the information from Abby and all of the information from Grover. It's only half of Abby's information and half of Grover's information. So that's a change. That's something that's different. That's going to result in an offspring that is different from either parent. And then we're going to move on to question number 5. And so for question number 5, I'm going to do some drawing here. And this is where we're going to talk about our some new information. And we're going to talk about Punnett squares. If you're in VLA, we've already talked about Punnett squares, so that you have a little bit more familiarity than the people in person do with this particular part of the video. But this is something that we can use as we're going through this question number 5 right here. So if the parents have another offspring, will that offspring be identical to the first offspring? Why or why not? So those other offspring are probably not going to be identical to the offspring that you created. So when we looked, for example, in class today at Zoe and Elmo, like I said early on in this conversation here, when we looked at those two, people had different outcomes. Their offspring that each of the different people stood up and shared about, they were different offspring from each other. They weren't all identical. So how could we predict that? Well, that's where this idea of something called a Punnett square comes in, and that's where the Punnett square is really, really quite helpful. So what we talked about in class, I'm going to write this in as Punnett squares. So what we talked about in class is that we can go through the whole process of meiosis, but if we're going to go through the whole process of meiosis, that's kind of a long process. And I'm gonna take you over here and show you this. We're gonna start talking about pea plants for just a little bit because we really need to move on away from our Sesame Street characters and understand that this all exists out in the real world and people study this stuff. And so we're going to take a look here at pea plants. We've got 7. So 1, 2, 3, 4, 5, 6, and 7. We've got 7 chromosomes in pea plants. A pea plant will have 2 copies. So it'll have 2 chromosome 1s, 2 chromosome 2s, 2 3s, 2 4s, 2 5s, 2 6s, and 2 7s. So the pea plant will have 2 copies of each of those chromosomes. Um, and so it's going to have 2 versions of each gene. So when we're looking at flower color on chromosome 1, there will be flower color on this chromosome 1, and then on the other chromosome 1 that it has, it'll also have flower color right there. So um, two versions of the flower color gene. So I think we're going to take a look at pod color. And pod color is looking green is dominant, yellow is recessive, and we're going to use a capital G for our dominant allele, and we're going to use a lowercase g for our recessive allele. So I'm going to draw, let me switch back over here. I'm going to draw what this might look like if we were to cross 2 heterozygous pea plants. Okay, so I'm going to show you what it might look like if we cross 2 heterozygous pea plants when we're talking about pod color. So we could have pea plant number 1 right here with chromosome 5, 2 copies of it. Here is a big G, and there's a little g. And so this is chromosome 5. So let's say this is the mom. And we're going to say over here that this is the dad. There's really not a mom and a dad exactly in pea plants. We'll just go with it. So same thing here, heterozygous. This is chromosome 5. I'm only drawing in chromosome 5 because I don't need to really draw in anything else. So if we're talking about meiosis, if you're in VLA, you don't have to worry a whole lot about what I'm going to talk about here. But this is going through the whole entire process of how gametes form. You're going to get this again in high school. So if you want to follow along with what I'm doing here, that's fine. If you want to kind of just get the overall gist, that's okay as well. So the first thing that's going to happen is that we've got each of these chromosomes making a copy of itself. And that's going to happen over here on the dad cells as well. Okay. And then we start to have the divisions happening. So now this chromosome pair is going to go into one of the gametes. And this chromosome pair is going to go into the other. Law of independent assortment, doesn't really matter which one these go into. They just go into 2 different cells here. Then these 2 are going to split out. And we're going to get all 4 of the gametes form. If you're looking at this in VLA and going, we did this and we came up with like 1 gamete, how is this possible? We just really kind of skipped looking at all the different possibilities and we used the coin flip to come up with 1 possibility. We're just expanding out what we did in class. Okay, so here's the information for the mom. I'm going to pause this for just a second and draw out the other for you. Okay, now that we're back, you can see on here that the same basic thing happened with the dad pea plant. And I went ahead and put these in the opposite way, over here as over here, because I want you to see that it's not that this type of chromosome is going to go a certain direction all the time. Okay. When we look at these two gametes are the same. There's a big G, or a dominant allele. When we look at these two, they're the same, recessive allele. These two have a recessive allele. These two have a dominant allele. So those are the possible forms of the gene that can be passed on. Now, truthfully, we could have looked right up here and seen the same thing that we wound up with down here. But this is the process of how that happens. So this is the whole model of being able to see what the possibilities are within the gametes. So these would be egg cells, or ovules, ova, and this would be sperm or pollen. So we've got um, the two different types of sex cells. Okay, now this gets really complex to write out. And so, it's kind of nice sometimes when you have a shortcut. We all like shortcuts. So I'm going to show you a shortcut to be able to predict what the possible outcomes might be. You can look at the possible outcomes here. I'm going to make a line down through here. So you could say, well, the dominant allele and the recessive allele could combine, the dominant and the dominant could combine, the recessive could combine, or the recessive and the dominant could combine. And you could say, okay, yeah, well, those are all the possibilities. You can do all of this a whole lot more quickly if you use what we call a Punnett square. So I'm just going to draw a little square here in my notebook. And then I'm going to cut that square into fourths. That's a little bit off there, but that's close enough. And then I'm going to take the two possible alleles that the mom can pass on. And I can either get that from going through this whole process here, or I can just look at what her genotype is, because her genotype is heterozygous. So if I go, okay, well, the mom is heterozygous, I want to write the mom's information here, right there. And then the dad, he can pass on a recessive or a dominant. I could get that information from right there. We know he's heterozygous as well for pod color. So I want to write his possible gametes across the top. So he could have a gamete with a dominant allele. He could have a gamete with a recessive allele. The mom could have a gamete with the dominant allele or a gamete with the recessive allele. Okay. This is going to fill across. That's going to fill across. This one's going to fill down. This one right here is going to fill down. So we're just going to look at pulling this letter over here and bringing that letter down there. So one possible combination for the offspring would be homozygous dominant. We're going to pull this across, pull that down. We could have heterozygous as a possible result. We're going to pull this across and that down. Convention is that the dominant allele is always written first. So heterozygous. Pull that one across, that one down. This is homozygous recessive. So here is our other possibility, or our last possibility. Now you're going to notice that this one, so the homozygous dominant, and this one, the homozygous recessive, those are not going to look like the parents. So there's a good chance that you're going to end up getting an offspring that doesn't look like the parents, or doesn't have the same genotype, so they don't look genetically the same as the parents. So let's just kind of write out what the possibilities are. So we could have homozygous dominant, and there's 1 out of 4, or 25% probability that this would be the genotype of the offspring. We could have heterozygous, and that's 2 out of 4, or 50% chance that this would be the offspring. We have homozygous recessive. There's 1 out of 4. So that would be a 25% chance of homozygous recessive being what we see. These two genotypes result in a green phenotype, and this one results in yellow. So there's a 75% chance of having offspring with green pods, or a 25% chance of yellow. And if you had 100 pea pods from these two parents, so you just keep mating these two parents, which is completely and totally possible, you'd probably have about, like if you had 1,000 offspring or 1,000 pea pods, you'd have about 750 of them be green and about 250 of them be yellow. So what you actually see in real life is that these numbers actually do play out roughly roughly as predicted. Now, this is how you can answer this question. So, if the parents have another offspring, will that offspring be identical to the first offspring? Most likely no. And you can use a Punnett square to show possible outcomes for combinations of gametes for the future offspring that might be produced. So that is how you can use a Punnett square to answer this last question. Now, if you prefer, if you really like this whole process here, this is perfectly fine. This is correct. You can use this, and you can talk about how this gamete and this gamete could combine together and produce an offspring, or these two, or these two, or those two. So you could go through the same process with this model, or you can use a Punnett square and go through the same process with this model. This one's a little bit simpler. We've been doing this a little bit longer. It doesn't really matter. It doesn't matter how you go about it. Either way is perfectly fine. You can talk. You could even talk about how we did the model of flipping the coin to see which chromosomes would be passed on. So you could use that as the model that you talk about. The which model you use is not important. What matters is that you have a model that you can use to support your answer, that other offspring are not likely to be identical to the parents, that you're going to have genetic variation or a variety of genetics when you're comparing offspring from or with the parents, and you're looking at other offspring that those parents produce. Hope this is helpful. I know that it's quite long, but hopefully this will give you a good understanding of what we talked about in class today, and also a good review if you need this as a review. Thanks, and have a lovely evening.